Hey guys, today I will be sharing with you guys a paper published in Science back in 2010. This paper is almost a decade old now, but it's more relevant than ever in terms of creating a functional and safe model for pharmaceutical drug testing. So, for those of you who are unfamiliar with this concept of an organ on a chip technology, let me just give you some context, right? So, in 2010, a team of researchers and engineers at the Weiss Institute at Harvard University came up with this design of an organ on a chip. And it's exactly what it sounds like. It's a transparent microchip where they have introduced hollow microfluidic tubes that are cultured with human cells on different sides. And through these hollow tubes, things like air, cells, nutrients, blood, and even pathogens can be pumped through. This convergence of like microlabs on chips technology and cell biology has allowed them to mimic the architecture and function of an actual organ at a super tiny scale. Okay, but why should you care? Well, the current models for testing pharmaceutical drugs, right, are either animal models or cells in culture, and both of which are not great predictors of what actually happens in human organs. So, with this organ on chip technology, we're able to develop this human in vitro model for healthy or diseased organs. You can use it to investigate mechanisms underlying disease progression. It could be used for drug development and toxicity screening, and it could replace clinical trials because it's safe simulation. Maybe in the future we might even have personalized drug simulators, and so far scientists have got pretty much everything on a chip, and maybe we'll even have a human on a chip. So possibilities are endless. And this image right here just shows that we could have dynamic cues and mechanical distortions and biochemical stimuli when we've got these cell cultures on chips. So how exactly did they build this functioning lung on a chip back in 2010, you might ask? Well, they did so by reproducing the structural and mechanical properties of both the alveoli and capillaries found in the lungs. And they created this alveolar capillary interface, which is the smallest functioning unit of a breathing, living lung. So this is an alveoli, single alveolus. And they were able to also recreate lung responses such as pulmonary inflammation in response to an infection, and they also had some toxicology applications in responses to nanoparticles. So how are the scientists able to recreate something such as the alveolar capillary interface on something as small as a microchip? Well, they started off with creating two microchannels separated by an ECM-coated PDMS membrane, which was a flexible membrane, and then they cultured two different types of cells on either sides. So the human alveolar epithelial cells are just epithelial cells from the alveoli, and the endothelial cells were isolated from the pulmonary artery. Now, this compartmentalization made it possible to manipulate fluid flow and delivery of nutrients and such separately. Now, once these cells were grown to confluency, right, air was introduced to the epithelial compartment so that it would create an air-liquid interface, which is a more accurate representation of the lining of the alveolar interface. Now, during normal respiration, right, there's intrapleural pressure, which decreases and causes these alveoli to expand. And to mimic physiological breathing, the authors incorporated two lateral microchambers and applied vacuum to it to cause pressure-driven stretching. So these are the three layers. This is the epithelium, endothelium, and these are the vacuum chambers. And keep in mind that all of this is happening in a chip this small. And this is a video of the actual stretching, so you can see the mechanical elastic movements that are happening right here. So you might be wondering, how does this compare to in vivo lung function or other in vitro models such as cell cultures? Well, to validate their study, the authors introduced these alveolar epithelial cells and the endothelial cells into the system, which showed to attach to the opposite surfaces of the PDMS membrane, and they formed monolayers made of cells, and then these cells had junctional proteins formed between them. So the epithelial cells had occludin, and the endothelial cells had VE, vascular endothelial cadherin. So remember how I told you guys that the authors had introduced air into the epithelial layer, right? Now, they did a bunch of tests and found out that the air-liquid interface had stabilized the liquid layer, which was same in vivo, and there was no drying observed as in other cell culture in vitro models. And there was also an increase in electrical resistance as seen in this diagram right here, And there was a low level of albumin protein permeability, which was like 2% per hour, same as it is observed in vivo. Now, this is just a video that shows real-time cyclic stretching, and these are a bunch of fluorescent particles on top of the PDMS membrane. So the other thing that the scientists were able to do was they could replicate lung inflammation via infection, right? And the thing is, cytokines are produced by parenchymal cells of the lungs, which were obviously missing in this microsystem. So to cause the pulmonary inflammation, the researchers induced TNF-alpha stimulation, which is a pro-inflammatory mediator of the epithelium. And this resulted in an increased endothelial expression of ICAM1 within just 5 hours of addition. It also caused the recruitment of neutrophils, and both of these were missing in the controls. This is just a time lapse showing the E. coli getting engulfed by phagocytosis. Now, without going into too many details, I'm just going to let you guys know what the researchers found from their nanotoxicology study. And it seems that toxicity is correlated to movements of the system, or the lung, which is really cool. So in the absence of any mechanical disturbance, there was little to no ROS generation observed. But with just 10% mechanical strain, there was a steady increase by a factor of 4, which has obviously important pharmaceutical implications for nanoparticles in medicines and things. Now, these are all the things that we covered in this video, and I would highly encourage you to pause and check if you understand them. And if not, you can send me any questions that you have. And thank you for listening. I hope this was helpful.